All right, good, thank you. I must, of course, I owe this visit to the organizers, and I thank all of you and Roger for thinking of me. But before I start, I don't really have any jokes. I'm sorry. It's a good one. But every time I come to Jerusalem, I'm in awe because I think I know Jerusalem quite well. In fact, I took Tom for a little walk in the old city, and I was able to guide him. But every time I come here, I realize I know so little about it because there is so much to see and there is so much to do. So you should feel very proud of being in this wonderful city. And it's a bit like science. And I'm going to take that analogy to science, that you feel in science that you've done something and it's over. It's complete, it's accurate and permanent. And I think Thales, the great Greek scientist, said, I don't know how many thousands of years ago, that life science is never complete, never accurate, never permanent. As you observe something, as you discover something, you find there's a lot of depth which needs to be evaluated, and then you get on to doing even more complicated set of experiments. The reason I make this point is all about the fact that in 2013, Jim Rothman, Sheckman, and Tom Sudoff were awarded the Nobel Prize for vesicle-mediated protein transport and the timing of the transport event. And I was at a meeting with the two of the giants, Jim and Randy, and somebody asked, so should anyone be working on membrane transport protein secretion anymore? And so you can imagine the answers, the kind of answers that were exchanged, or the discussion that took place after this interaction. So I'm going to try to tell you a little bit about the history of protein secretion, and then I'm going to try to give you an idea or a feel for some of the big issues that remain unaddressed, and there's a lot of life left in this process. So if you're interested in protein secretion, keep at it. There will be rewards at some stage in one's life. So this, oops, here we go. So this slide, I stole this from George Pallady's Nobel lecture in 1974. And I used to talk to George because he was my colleague at University of California, San Diego for many years. So in his 1974 lecture, he basically presented data that proteins that are made in the endoplasmic reticulum, and you heard about that very nicely from Tom earlier today, that proteins that are made in the endoplasmic reticulum, and now we know that a third of the genome encodes for proteins that are inserted into the ER or translocated into the ER. There's a lot of proteins. Many of those proteins, of course, remain in the ER to serve the functions of protein folding, quality control, etc., etc. But then a vast majority of those proteins are exported and secreted outside of the cell. So that's what George was interested in, trying to elucidate the mechanism of how those proteins go out of the cell. And what he did was he was able to provide a map, as it were, of the path taken by proteins upon their export from the endoplasmic reticulum and before they come out into the extracellular space. And what he said was that proteins start their life in the endoplasmic reticulum. They are then carried to a compartment called the Golgi complex. And from the Golgi, the cargoes are collected into a large container, or a condensing vacuole, as he had called that in that year. And these containers then ultimately fuse with the cell surface to deliver the cargo. So he came up with this scheme that compartments of a eukaryotic cell maintain identity by exporting material that is required at the next station and retaining those that should only function at the maternal compartment. So this way, not only you maintain the identity of the individual compartments, but you also deliver cargoes to the right place. So this was his Nobel lecture. And now over the years, so that was 1974, so you could basically explain his working model by just looking at this cartoon. So what he's trying to say, or what he said, was that green molecules are to be taken out from this particular compartment and delivered to this particular structure. So this would be the departing station, and this would be the arrival station. And what you do is you collect, I'll run it again. You collect the cargo in green in small containers, and you leave behind the resident in red. And these containers arrive at the target membrane, they fuse and deliver the content. This is a very simple principle by which proteins are secreted. Now, over the last 40 years, we've learned a lot about this process. We have a very good understanding of how proteins are trafficked from one compartment to the other. And I just listed some of the key players here. But there's a lot of contribution by many others. So Blobel basically came up with the scheme that proteins enter the endoplasmic reticulum through the presence of a signal sequence, which Tom described earlier on in his talk. So the proteins get into the endoplasmic reticulum, and then Jim Rothman, Randy Sheckman, and Tom Sudoff, and their labs and many other labs that are not listed here, they were able to provide details on how the cargoes are captured into vesicles. So any cargo that needs to leave the endoplasmic reticulum is captured into a COP2 coated vesicle from the ER. These vesicles travel in the direction of the Golgi, and once the cargo arrives in the Golgi, they are picked up by COP1 vesicles, which in fact I had purified in 1989 in Jim Rothman's lab. These vesicles, when we purified, we were absolutely sure that they're going in this direction, meaning moving away from the ER in the direction of the cell surface. But the data that we have now suggests strongly that these vesicles are bringing material from the Golgi back into the ER. So when you go in this direction, you also need to bring components that went in this direction. You want to bring them back so they can be used. So COP1 vesicles are likely, we know for sure that they are definitely involved in this pathway. Whether they play a role in this pathway is still controversial. Now here's something worth remembering. Till about 1970s, people were not even sure if Golgi complex is a real compartment, right? So, I mean, this area of biology has been filled with controversies. So there's nothing new now in terms of controversies. So, whether the trafficking of cargo in this direction is by means of COP1 vesicle or another kind of vesicle, or by a process that is generally called cisternal maturation, meaning you don't really have vesicles carrying cargo in the forward direction. What happens is these cisterna keep moving forward, and whatever should not go in the forward direction is being taken out, as it were, and recycled. Okay, and it's going to take a few more years before that situation is solved. Anyways, once you get up to the very last compartment of the Golgi, containers then collect cargoes from here and bring them to the cell surface. The identity of the containers is not very clear. We've purified one class of those vesicles in my lab, and we call them carts. Okay? But there are likely to be many, many different kinds of containers that bud from this compartment and take cargoes to the endosomes, to the apical surface, to the basolateral surface, and now it turns out that the plasma membrane here is in fact made up of specific domains, and it is very likely that they receive cargoes from the Golgi by means of specific vesicles, right? So clearly how cargoes are sorted and packed at the ER and then delivered to the respective destination remains a major challenge. Okay, so fine, we now know that COP2 vesicles go in this direction, COP1 vesicles go in this direction, and perhaps also in this direction, and there are issues with respect to how cargoes go from Golgi to the cell surface. And this is just to give you an idea of what COP2 vesicles look like. We call them COP2 because they were purified in 1992 in Randy's lab, and COP1 vesicles were purified in 1989. Okay. So COP2 vesicles, this is what they look like by microscopy. And they have a size which is about 60 nanometers in diameter. Okay. These vesicles have been purified. We know their polypeptide composition. We know a lot about how these vesicles form. Okay. But there's a very big problem that has remained unaddressed in our field. And that has to do with the fact that higher eukaryotes, cells that you and I are made of, flies, worms, etc., fish, etc., they make and secrete proteins called collagen. So in the humans, there are about 29 different kinds of collagens, and there are many collagen-like proteins. And you absolutely need this collagen. So without collagens, you don't really have skin, you don't really have bones, you don't have tissues. Okay? And to date, there are close to 1,400 mutations that have been mapped to collagens. And any mutation in collagen usually leads to at least a defect in skin organization. But there are very, very severe forms of human pathologies associated with collagen mutations. And the problem is, collagens are far too big to be contained in a COP2 vesicle. So if you look at collagen type 7, which you absolutely need to assemble skin, this collagen has a very rigid triple helical region, which is 450 nanometers in length. So it is a long rod-like structure. And there is no force inside the cell that could bend it. So there's no Arnold Schwarzenegger inside the lumen of the ER that could bend it into a structure that could be accommodated into a 60 nanometer COP2 vesicle, right? So these proteins are made in the ER and they follow the same route of export that I told you earlier. So how would a cell then export collagen type 7? Collagen type 1 is slightly smaller. It's about 300 nanometers in length, okay? Now, in addition to the fact that they're very important for skin biogenesis and bone structure, et cetera, you'd be surprised to hear that 25% of your dry body weight is collagens. That's a lot of proteins. And yet, our understanding of how these proteins are secreted remains, let's put it this way, it's not satisfactory. Okay? The reason is because in our field, there were two approaches that were used to understand protein transport. One was Jim Rothman's in vitro reconstitution. Brilliant approach, okay, which led us to an understanding of the mechanism by which you form vesicles and you fuse them. And the second was Randy Sheckman's secretion mutants. This was performed in the year 1977, but he used Saccharomyces cerevisiae as a model system to study protein secretion. And yeast don't make skin, they don't make bones, they don't secrete collagens. In fact, yeast don't really secrete that much to begin with. So you were not likely to, you would not understand how collagens are secreted by studying yeast as a model system. So this basically issue had been left sort of unaddressed. People just ignored it. Whenever the topic came up, so how do you explain collagen export? You know, the experts basically just said, wow, we'll get to it. You know, but the point is, we need to get to it now because we understand a lot about protein secretion in general. So this is just to show you a gory picture that, defect in secretion of collagens or in mutation results in collagen 7 not assembling into ECM, and you get this terrible human pathology called epidermolysis bullosa, and there's no cure for it. I'm going to just jump to the next one. And there is no cure for it because you can't really do anything about it. So there is therefore a challenge for us to understand how are bulky collagens exported by cells. So before I get to that, I need to tell you something about the mechanism of COP2 vesicle biogenesis at the ER, because this is very important. This is the foundation on which we are going to build the whole tenet of how collagens are exported. So this process requires 6 proteins. Okay, what are these 6 proteins? So there is a GTPase, a small GTP binding protein called SAR1, which lives in the cytoplasm in a GDP bound form. When it interacts with a transmembrane protein at the endoplasmic reticulum called SEC12, the GDPs change for GTP, and when that happens, SAR1 inserts into the outer leaflet of the endoplasmic reticulum. Once that insertion has taken place, it then recruits 2 proteins, SEC23, 24, as shown here. SEC23 has the ability to hydrolyze GTP to GDP, but it does so very, very slowly. That GTPase activity is enhanced upon the binding of 1331 to 2324. So you should think of this process as initiation of vesicle biogenesis process, which begins by recruiting SAR1 to the ER. It then recruits 2 proteins, which we will call, for the sake of simplicity, the inner coat, followed by an outer coat, which is made up of 1331. Now, what it does is, this is to the best of our understanding. It's possible that, this is all you need. but I'm always the kind who is not easily taken by just what is present. It is possible there are surprises in this pathway as well. So what is being said is that a flat membrane like this, when it binds to these proteins, it does this, and then it pinches off. Okay? So a flat membrane does this through the function of these particular polypeptides. The function of this 1331 is then, the moment this complex associates with 2324, SAR1 GTP is hydrolyzed to GDP, and that terminates the reaction. So you need to convert GTP on SAR1 to GDP in order for a structure which grows out of here from the ER to separate from the maternal compartment. Okay? So one way of thinking about collagen export is that all you need to do is come up with the mechanism, because come up with two sets of components. One would be, so collagens are proteins that do not have transmembrane domains, except one, collagen 17. Okay? So Sec24 in this complex is the molecule that binds to receptors. The receptors can then pick soluble proteins which do not have access to these coat proteins. Okay? So for collagen to interact with these coats, you need to invoke the presence of a collagen receptor, which would connect collagen in the lumen of the ER to these coat proteins. And then you need to come up with a mechanism by which you increase the surface area of the endoplasmic reticulum. Because if you imagine that in order to make a vesicle of 60 nanometer, what you might do is on the surface of the ER, you would spread a coat which could be 100 micrometers by 100 micrometers, which would do this and give you a structure which is 60 nanometers. Okay? But in order to make something that is really large, in order to accommodate something that's 450 nanometers, you need to provide a lot of surface area. So all you do is you make a bigger coat. Okay? And so instead of using a handkerchief to pick something, what you do now is to put a blanket, as it were. Okay? But that depends on the following assumption, that coat proteins are required for collagen export. And this has been challenging because there were reports till about 8 or 9 years ago that the export of collagen is independent of coat proteins. Okay? So how do you get to this process? So, yes, sir. So, yeah. Nothing, you don't need anything for COP2 coats. For Clathrin-coated vesicles, you need oxalin and HSC70, but for COP2 coats, you don't. But that's the current thinking. There was this idea. Sure, trap complex, right. But there's a bit of controversy there. Before, the idea was that the moment vesicle comes out of the ER, it sheds its coat. But now, according to Susan, the coat remains on the vesicle till it comes to the Golgi. So that has to be resolved before you can give any weight to this whole idea of what is involved in uncoating. So it isn't clear. Yeah. So the minimum requirements for collagen export then are, you need to come up with a receptor that binds and sorts collagens, and you need to come up with a mechanism by which you increase the surface area to generate a bigger membrane-bounded transport carrier. Okay, so how does a cell do it? So I'm going to cut a very long story short. Fred Bard, when he was a postdoc in my lab, identified in the year 2006 a large number of new genes which were required for transport and Golgi organization. So this was a screen we performed in Drosophila S2 tissue culture cells. I think we were the first ones to perform a genome-wide screen in metazoan. And we identified 74 new components that had not been given the function in protein secretion or in the organization of the Golgi apparatus. And the reason why I'm mentioning this is because how a Golgi is assembled and why does it have the shape it does is something it is of great interest to me, and my lab has worked on it for years, and we're still not quite there yet. Okay, so obviously we are interested in identifying new components in this pathway. So for the sake of simplicity, we came up with an acronym TANGO. So these are TANGO genes, and since 2006 we've been basically trying to address the function of these TANGO genes in protein secretion. And I'm going to tell you the function of one such protein called TANGO1. So Tango 1 is a protein of 1907 amino acids. It sits at specific sites at the ER, and for the sake of simplicity, these sites are called ER export sites. So they are very much like departure gates at airports, where cargoes, well, at the airport you will have passengers, they come and they're checked and then they're asked to go and board the plane and the plane takes off, right? So those are departure gates. So similarly, you have at the ER these so-called departure gates, but the composition of the departure gates in terms of molecules, lipids and proteins, which separates it from the rest of the ER, the non-departure gates, is not clear. So anyway, Tango 1 sits at the ER exit sites, and it has the following topology. So it has 900 amino acids in the lumen of the ER, and at the very tip of the molecule is an SH3-like domain. There's a coil-coil domain in the lumen of the ER, and there are 500 amino acids which have no structure, okay, that we can predict based on what's available to us. On the cytoplasmic site, it has 2 coil-coil domains and a proline-rich domain at the very tip of the molecule. It has a very peculiar topology. It has 1 full transmembrane domain, and it has a hairpin loop. This hairpin loop is found in proteins that Tom has worked on for a large number of years, the proteins called reticulons, for example. And we believe that this domain plays a very important role in the function of the protein that I'm about to tell you. So when we observed this peculiar topology of the protein, we became excited about working on this particular component. And Kota Saito, when he was a postdoc in my lab, took the challenge of developing reagents to understand what is the function of Tango 1 in protein secretion. So this is just to show you what an ER exit site would look like in a mammalian cell. So you stain a cell with an anti-tango antibody, and what you find, so this is where the nucleus would fit in this particular cell, and you see punctate staining, puncta, spread throughout the cell. And so these are the hallmarks of an ER exit site. So this is where cargoes are collected, and this is where ordinarily COP2 vesicles will pinch and leave in the direction of the Golgi. And this is just an inset to show you one such area that they are separate dot-like structures, okay? So Tango 1 is an ER exit site protein. So when Kota, in order to understand its function, Kota found something very peculiar. He found that Tango didn't seem to have a role in general protein secretion. So if you were to look at proteins such as albumins and antibodies that are being secreted by cells, it didn't really seem to, they don't seem to require the function of Tango. So you could remove Tango. This was done before CRISPR. We were using shRNA and siRNA-based knockdowns of Tango 1. It didn't seem to affect the export of many of the cargoes we'd looked at. So we were flummoxed for a while as to what on earth is the function of Tango 1 at the ER. So Kota did what any biochemist would do, and he immunoprecipitated his protein with the antibody he had generated, and we gave it to the mass spectrometric facility at the CRG. And we came up with results, and we found that the IP contained 6 peptides corresponding to collagen type 7. This was a bit of a surprise, a pleasant surprise, I should say. So we then reasoned that perhaps it has a role in export of collagen type 7, or perhaps the physiology of collagen type 7. And I think it's worth mentioning here that collagens, there are something like 80 to 90 proteins that are dedicated to the folding and assembling of collagens before they leave the ER. This is a very complicated assembly process. So we thought, well, perhaps it has a role in assembly reaction. So turns out when Tango is, Tom is communicating my talk to my competitors, right? So Kota then knocks down Tango 1 in cells that secrete collagens, especially collagen type 7. And it was very easy for us to then conclude, based on the data we had, that when you remove Tango 1 by siRNA, there's a defect in the secretion of collagen type 7. As shown here, there's about an 80% reduction in the amount of collagen type 7 upon knockdown of Tango 1. And there is a concomitant increase in the amount of collagen that can be seen inside the cell and inside the ER. So that gave us the confidence that Tango 1 had a role in collagen type 7 export. Okay. Good. So This is one of the things that I always tell my lab members and anyone who's interested in listening to what I have to say, that when you do these kinds of experiments in tissue culture cells, you come up with data and you propose an idea, propose a hypothesis, but that's not in vivo. That's in just tissue culture cells. So you have to be able to then go and test the value of your in vitro finding data by doing something that takes you as close to physiology as possible. So we were trying to do the Tango-1 knockdown with my colleague Reinhard Fassler at the Max Planck Institute in Munich. We were very slow. He's an expert, but we are not so good at generating mouse knockouts. But it turns out Genentech did the work for us. So Tango has been linked to atherosclerosis because of its role in collagen export and something else that I'm about to tell you. So they had made a mouse knockout of Tango-1, and what they found to their surprise was that the pup is born. So this is the wild type pup, this is the mutant pup. It's born, but it dies rapidly or soon after birth. It dies soon after birth because it doesn't really have any skin, and the bones are like rubber, so it cannot suckle it. And in fact, many of the bones are missing. It doesn't have the lower jaw, it's missing the clavicle, and so it's in very, very bad form. And then they went and looked at, based on our story that we had published 2 years before this date, they went and looked at the export of collagens, and they found that Tango was in fact required. Tango is also called MIA3, so you needn't worry about that. What they found was that Tango-1 was required for the export of collagen 1, 2, 3, 7, 9, 11, and many others. Okay. So this therefore gave us the confidence at two levels. One, that what we had said based on our tissue culture-based analysis held some value. Okay. Number two, we were just gung-ho on collagen type 7, but this told us that it is likely to be required for export of many different collagens, and that made more sense and made it a little bit more exciting to work with. So with this level of confidence, we then decided we should continue with our studies, and now comes the difficult part. Getting the molecules was not easy, but sure, you can get to it. But the mechanism is where you come up with a hypothesis, you test it, and you find, you've got more work to do. So I'm going to tell you our current understanding of this process. Okay? But this is something that Tom had said in his lecture, that what he had proposed many years ago was right, except one or two things, right? So I'm going to tell you that the model we proposed in 2009 stands correct up till now, till we are proven wrong. Okay? No, but it's not just by us, by the others as well. So I think that's the important thing to remember. So how does Tango do it? So I'm going to tell you that Tango acts as a scaffold at the ER exit site, and it recruits at least 3 different classes of molecules. And in doing so, it prepares the endoplasmic reticulum to generate a mega transport carrier to export collagens. What are the molecules? Through its proline-rich domain, it interacts with the inner coptocoats, that's Sec23, Sec24. Through this SH3-like domain, it interacts with cargo, the collagens. And this particular domain of 138 amino acids proximal to the ER membrane acts as a tether for recruiting membranes, which I'm going to show you are used to generate a mega transport carrier. Okay? So how does it work? Or how did we get to this stage of analysis? So the first one, Tango-1 binds to Sec23A. So Sec23A, Sec23 is the GTPase that I mentioned in my introduction. Okay? And it's mediated through the interaction with the proline-rich domain. So when Kotha was in my lab, he performed a very, very simple experiment, yeast to hybrid. And he asked what can be deduced from a yeast to hybrid based analysis to ask whether proline-rich domain had a functional significance. And what he found was that this particular region here, which is the proline-rich domain, interacted with Sec23, as shown here. So he's looking at this molecule and this molecule. So we just focus on C2. This CT2. CT2 domain interacted very, very strongly with Sec23A. So based on the analysis just of this kind, we proposed that Tango-1 was interacting with Sec23A. More recently, we've been able to do this biochemically. You purify proline-rich domain, you incubate with Sec23A, and you ask whether they bind, and the answer is yes. But we were saved, or I should say we we were helped in this process by a paper that appeared earlier this year from Jonathan Goldberg's group. He's at Sloan Kettering in New York City. What he did was he in fact identified the domains within proline-rich domain of Tango1 that interact with Sec23. So he could co-crystallize the two proteins. And he found that proline-rich domain of Tango contains small subdomains, which are basically 3 prolines here. So there are 7 of those. So this is proline, 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 and it is repeated 7 times. And it is these prolines that bind to a gelsolin-like structure in Sec23. So he solved our problem by doing this crystallography. I don't think we would have been able to do this, but he's done it. It's a great support for what we had said almost 5 years before this paper came out. So this tells us how Tango would interact with Sec23. Now, Tango binds cargo. So I'm going to show you that this interaction takes place through the SH3-like domain. So how did we do that? So Kota, when he was in the lab, he performed a very simple experiment. He expressed Tango1 in cells, immunoprecipitated it, and incubated it with pure collagen type 7. So we could express full length, the luminal domain only, the cytoplasmic domain only, and a protein that is full length, except it doesn't have 48 amino acids that correspond to the SH3-like domain. And all you have to do then is to ask which of these molecules can bind collagen. So the full length binds, that's great. The luminal binds, that's very good. The cytoplasmic does not bind, that's brilliant, because collagen should be in the lumen of the ER, not in the cytoplasm. But most interestingly, a full-length protein that does not contain the 48 amino acids does not bind, or binds very weakly to collagen type 7. Okay, fine. It turns out, again, Hans-Peter Backinger, who was at Oregon Health Sciences University, published a paper just about 2 months ago in which he showed that the SH3 domain of Tango binds to HSP47. HSP47 is a common chaperone that binds to all the collagens. Remember, we had only looked at collagen 7 binding. We could not explain how Tango would bind collagen type 1, collagen type 2, 3, 4, 5, etc., etc., etc. But this solves a big problem that we had been dealing with for a number of years. So it turns out HSP47 would bind to all the collagens. This is known. So by binding HSP47, what Tango is doing is it's recruiting to itself, as it were, all the collagens that are known to be made in the lumen of the ER. So this solves another very big problem for us, that this is the molecule, this is the part of molecule Tango1 which interacts with collagens, but it does so through HSP47. So collagens have weak affinity for Tango1, but this affinity is increased tremendously. I don't have the exact numbers, but the paper came out in PNAS just about 2 months ago, when HSP47 is bound to the SH3 domain. So this solves that problem. So based on what we had published in 2009, before knowing the work of Backinger and Goldberg, we had proposed the following model in 2009. So we proposed that ordinarily Tango is sitting at the ER exit site, and it has no role that we know of per se in general protein secretion. But when collagen is made, it binds to Tango, and the binding induces a change in conformation in such a way that the proline-rich domain here now interacts with Sec23. When Sec23 is occupied by Tango's proline-rich domain, Sec31 cannot bind. Why? Because Sec31 contains a proline-rich domain which is required for its binding to Sec23. So it is as if the plane comes in, but it cannot get to the docking site because there's another plane parked. Okay. So it's exactly, it's a similar, it's a sort of silly analogy, but it's, this is what I'm trying to get at. That Tango binds to this protein and prevents the recruitment of the outer coat. And we proposed in 2009, based on this, that once Tango is bound to Sec23, which takes place only when it's bound to collagen, it's pushing collagen from the luminal site and it is holding on to Sec23. And what it does is it allows the cells to make a bigger structure. Why? Because the membranes keep growing. In order for this reaction to terminate, the cells have to recruit 1331. But because that site is occupied, 1331 cannot come to 2324. Therefore, the structure keeps getting bigger. Okay? There will come a time when the cargo dissociates from SH3 domains, because it's going to try to push it in this direction. The binding affinity of SH3 like domains to the cognate clients is weak affinity. There will come a time when they will separate. Or if you want to make life more complicated, you could invoke the involvement of a new component in the pathway that would separate the two. Okay? Once that separation takes place, the proline-rich domain separates from 23, which allows 1331 to bind, which then hydrolyzes GTP, and the structure separates by membrane fission, thereby giving a bigger transport carrier. So, yes? It is.
No, what prevents the outer coat from being connected to the inner coat if triangle 1 isn't covering the entire budding
Yeah.
when it is still growing?
I'm going to tell you that. Just give me a. I mean, this is part data, part fiction. But I'm going to tell you that I think what we said in 2009 seems almost correct. So, anyone else? Yes.
People really identify that as deep.
Remind me again towards the end of the talk, because I will come to that, because that's a very important question.
And these threads would just contain one molecule?
Oh no, we have no idea. But I think we calculated with Matthias Mann as to how many molecules, and we came up with a number that it could be something in the order of 100 primers of collagen per transport per ER exit site.
Like
What?
The purely collagen.
We don't know. Again, this is to do with the question you're asking, whether collagen-containing carriers have been isolated, visualized isolated, and do we know anything about how much cargo is contained and what kind? Are they pure collagen or are they mixed in content? We don't know that. Okay, so this is what we proposed in this particular year. A very simple model to explain how collagens are exported by a tango-dependent reaction. So at least this part was clear, that this acts as a receptor or a scaffold that binds and sorts collagens. Okay? Now the question is, how do you increase the surface area in order to generate these big structures that you need to put bigger, bulky cargoes? So one very simple assumption would be, so as I said earlier, that in order to make a 60 nanometer size vesicle, perhaps you need to assemble a structure of coats which is only this much in length, and of course you have to think in 3D, so it'll be structure like this. So okay, but if you want to make a much bigger transport carrier, what you might do is, or the cells might do, is just generate a much bigger coat on the surface of the ER. So if this gives you this is bigger, therefore it gives you a bigger transport carrier. And we thought, let's test this. So we could visualize, so for example, there are conditions, so collagen is in red here, and this is at the ER, okay? And the green are COP2 proteins that are visualized with an antibody. So here is a large patch of collagen in red, and what you see is that the coats seem to be in small punctate elements. There isn't a sheet of coat that covers this structure. Okay? And this is an electron microscopy based analysis of not the exact patch, but a similar patch. So here you can see with immunogold labeling, this is a patch of collagen. Okay? And what you see here are a collection of vesicles that are in the vicinity. So this experiment told us that we cannot see a large sheet of COP2 coats on the surface of the ER, which could help generate a big transport carrier. But what we saw repeatedly were these collections of vesicles here. And we reasoned that perhaps it is these vesicles that are fusing with the membrane to generate a big transport carrier. And I'll try to explain that more clearly soon. Okay? So here is the idea. So instead of assembling a huge coat structure to generate a big transport carrier, what the cells are doing, they are recruiting pre-made membranes to the site. So it is, if you were to go to Tel Aviv beach and you sit there, if you want to make a mound of sand, one way to do this is to just collect sand and make a bigger mound, right? So you go from flat to something like this. The alternative is You pick wet sand and you drop it. And as you're dropping it, it sticks and it grows in this direction. So I'm going to show you data that it is this idea, this model, and not just simply an increase in the size of the coat. So how do we get to that? So we found, this is Antonio Santos in the lab, who found that under many conditions where collagens are being transported from the cells, we could visualize patches of collagen as shown in red here at the ER. And we also found that these patches were studded with membranes that contain this particular protein, ERGIC53. So what is ERGIC53? ERGIC53 is a protein that starts its life in the ER. It goes as far as a compartment which is called ERGIC, which is short for ER Golgi intermediate compartment. And then it comes back. So it's cycling between ER and a station before the Golgi complex. Okay? So it is doing this all the time. And we found that it was attached to patches of collagen, as shown here and here. If you knock down Tango 1 by RNAi, and we have become smart now, use CRISPR-based technology, you knock down Tango 1, of course collagen is not going to leave the ER, it's arrested in the ER, but what we found to our surprise was that these ERGIC53 containing membranes had dissociated. They were not attached to this particular structure. So we reason that it is perhaps these membranes that would be fusing to a patch of collagen, thus giving rise to this megastructure. Now, so Antonio, in fact, was able to identify a small domain of 138 amino acids here, which was responsible for recruitment of these ERGIC membranes. Again, I'm not going to show you the data. It was published about a year ago. The beauty of this experiment is that you could take this patch, this domain of ER, this domain of Tango, and place it at the mitochondria. The ERGIC membranes go to the mitochondria. You could place it at the plasma membrane. The ERGIC membranes go to the plasma membrane. So that's very simple way of showing that this domain has the ability to recruit ERGIC membranes. Okay? And we called this domain TEAR, which is a good acronym for tether for ERGIC membranes at ER. So this part can recruit ERGIC membranes, but do those membranes fuse with the ER in order to generate this big structure that we are proposing? And the answer is yes. So when Cristina Nogueira was in my lab, she had in fact demonstrated that membrane fusion was required for collagen export from the ER. Now this is a bit of a surprise. Remember, membrane fusion, what is membrane fusion? A vesicle leaves a compartment, it comes to the target membrane, it fuses through Jim Rothman's famous snare-mediated principle, right? The membranes fuse and you get delivery of cargo from here to the target membrane. We found that membrane fusion was required for the budding event, which is where you start a vesicle formation process. So how did we get to this? So when Patrick Earlman was in my lab, he had taken up Kota's immunoprecipitates and he was sequencing and doing cleaner experiments. And he had found that Tango 1 co-precipitated a protein that is known in our field as SLY1. So this was the peptide that was identified. So what is SLY1? SLY1 is a protein that functions at the fusion event. Its exact role is unclear. It may have a role in organizing the snares in a way that they become fusogenic, or it could be regulating the number of snare molecules which are necessary to fuse membranes. But regardless, this protein only functions at the fusion event as far as the fusion specialists are concerned. So we found that Tango, which has a role in budding, co-precipitated SLY1. So if SLY1 is required for fusion and for collagen export, we should be able to show it. So when Cristina performed the experiment, so again knocking down SLY1 from cells, shown here, resulted in inhibition of collagen export from the cells compared to control. And this is when you knock down Tango 1. Okay? And whatever does not leave the ER is arrested in the ER. Okay? So this told us that SLY1 was required for collagen 7 export from the cells, just like Tango 1. Perhaps not as effectively, but it clearly had a role. So this is about good 70 to 80% reduction in the amount. Now if SLY1 is involved, then we should be able to identify the snares involved in this reaction. So yes, we can. So there are many snares between ER and the Golgi. So you have to be thinking about two classes of snares. One that's on the vesicle called a V snare. the others on the target membrane that are called T snares. And you need to have 3 T snares and 1 V snare in order for membranes to fuse. So what Ishii Rawate in my lab found was that of the many T snares in the ER-Golgi pathway of protein secretion, knockdown of BNIP1, syntaxin 18, or USE1, either of these 3 snares affected secretion of collagen, okay, compared to the control one. Tango 1, of course, is required for secretion. We've known it for many years. Syntaxin 5 is a T snare that sits at the Golgi. So if you don't have T snare at the Golgi, of course you're not going to get secretion, right? Because collagens have to go through the Golgi in order to be secreted. So this gave us these 3 T snares. So if there are T snares, there must be a V snare. So Ishii was also able to identify the V snare, and it turns out of the V snares, he tested only YKT6 knockdown affected collagen export. So this told us that SLY1 in association with 3 T snares and a V snare YKT6 was required for collagen export from the ER. So again, when you propose these kinds of data, there's always this little doubt in the back of your head that are we right? And it turns out that a paper appeared very recently, while it's still in press. Corrected proof, no 2 uses. Well, these good people wrote to me to tell me that they support what we had said 2 years ago. So what they found was that the SLY1 ortholog in zebrafish is called SCFD1, along with syntaxin 18, which we had shown, facilitates trafficking of collagen type 2. So they only looked at collagen type 2 in a tango-dependent manner from during development in zebrafish. So they provided the in vivo proof, which completely supported what we had said based on tissue culture-based analysis. So this therefore gives us the confidence to propose then that this part is really then recruiting membranes which fuse to the ER to generate a big transport carrier. So this part recruits SEC20, this interacts with SEC23, this with cargo, this one is recruiting membranes that fuse. So based on this, then we can come up with a working hypothesis. So the hypothesis is as follows. So this is the endoplasmic reticulum membrane. This is the lumen of the ER. This is collagen. This is Tango 1. This is SEC23, SEC24, and these are the collagen primers. So what we proposed in 2009 is the same here, basically that Tango binds to collagens. Of course, there is a change now that it is doing so through the function of HSP47, which would be here, and it's pushing. And one possibility is that as this is being pushed, this hard rod-like structure might then be able to create a small dimple in the ER membrane. Okay, so this dimple is coated with SEC23 and 24, and Tango 1 is bound to SEC23. This structure then grows. This is hopefully going to address the question you had asked. So this structure then grows through predominantly the push of collagen through the function of Tango 1. But it can only grow to a certain size. The reason being the following. Tango 1, when the collagens are leaving the ER, Tango does not go with the cargo. It stays there. Okay? It stays in the ER. And in order for it to stay in the ER, it needs this hairpin loop that I had mentioned earlier. Okay? And we believe that this structure cannot be more than about 40 to 50 nanometers. Why? Because Tango has 2 coil domains. And if you calculate based on the number of amino acids, then Tango can only stick out here to about 40 to 50 nanometers. This part, therefore, is not likely to be coated by Tango. Okay? So how do you then grow this structure further in size? Because collagens need something bigger than 450 nanometers. And this is only going to give you a tenth of what is necessary. How do you do it? But before I get to that, this hypothesis dictates that Tango is likely to be forming a ring, as it were, at the base or the neck of the structure that is growing out, and COP2 coats are going to be in the middle, okay, based on this drawing, right? So it turns out that is absolutely true. Ishii Rawate in my lab found by super resolution microscopy, again looking at the organization of Tango 1 at the surface of the ER and trying to understand its organization with respect to COP2 coats. And you can see that Tango in green, COP2 in the middle in red, appears to be in the form of rings. Okay. You could argue, well, they're not absolutely perfect rings. We don't know that, but it clearly appears to be forming a ring-like structure, and COP2 coats are in the middle, which would fit very well with what we had proposed, that Tango is going to be a ring going around the neck of this structure, and COP2 coats are going to the middle, and collagens would be in the center. Unfortunately, we cannot look at collagens, Tango, and COP2 coats all at the same time. Okay, we're trying to resolve that issue. Okay, these rings seem to be around 300 nanometers in size, but remember, this is using an antibody, so there is going to be some level of exaggeration in the size of this ring. It's likely to be something like 200 nanometers in diameter. Okay, so the idea is Tango forms a ring, and then I've told you that it has a domain which can recruit these ergic membranes. So these membranes are being recruited. As these membranes are being recruited, these membranes are prevented from basically merging with an ER in such a way that they just flow out. We think that this ring organization of Tango concentrates these membranes into this neck-like region, and the addition of these membranes is then allowing this structure to grow, as shown here. And once the structure has grown to a size that's big enough to encapsulate collagens, Tango separates from collagens. When it separates, it's here. The proline-rich domain separates from Sec23. When that separation takes place, 1331 become associated. Once that has taken place, now you're ready for cutting the membrane here. And that happens once the membrane is cut, a transport carrier is separated from the maternal compartment. Now, you asked me a very important question. Have we seen collagen-containing carriers? Well, people in our field have tried for the last 15 years, but no one's been able to see collagen-containing carriers separated from the ER, somewhere between ER and the Golgi. Why is that the case? Well, there are all kinds of excuses. Okay? So there is one group at the NIH who believe that they have seen collagen-containing carriers. But I'm going to suggest something. Randy and Rothman are not in the audience, I can say this. So one possibility is this, and I wrote this in a review, so it's been said. So one possibility is that you generate this big structure, right? What happens is this big structure, as it is growing from the ER, it has the capacity to fuse with the Golgi. So here is the Golgi. So you can imagine that this tip attaches to the Golgi through lots of tethers, which we have identified now, and it fuses this tip to the Golgi. Once this fusion has taken place, you have now a structure which is like a conduit, a canal between ER and the Golgi. Calais is connected to England through the tube, and the trains are going through. So the train in this case would be the collagens. And then all you have to do is you terminate the reaction, you cut it here, and the membrane is simply absorbed into the Golgi. So instead of having a transport carrier as defined by George Palladio, meaning something that is separated from the maternal and compartment and the arrival station, what you might have is a tunnel which allows cells to communicate from the ER directly to the Golgi. But then you would ask, well, things would leak out. You'll be in trouble. Well, if they leak out, they can always be brought back by COP1 vesicles. Yeah? So there is that possibility, and we are trying to test this experimentally. But we haven't reached there yet. The problem is it's very hard to label collagen with GFP and structures of that kind. So doing in vivo live cell imaging becomes difficult. Sorry.
Say they might come back. So this suggests that the Tango 1 is found also in the ER?
Tango 1 is always at the ER. It never no, it never goes to the Golgi. It never leaves the ER. It just stays there. It's
so is the mechanism for delivering them back to the ER known?
no, Tango doesn't have to leave, right? All you have to do is, once this separation has taken place, then you fuse. So this guy just stays here. This structure fuses, and the collagens are the ones that are going. So you're letting the cargo go, but you remain where you are.
Yeah, but I mean, first of all, the cargo needs to reach the Golgi.
Yes.
Right? And then it needs to be secreted.
Yeah.
So I believe that such mechanism will happen also in the Golgi.
Okay, okay.
For secretion.
Ooh, okay.
So
So
I'm asking
if Tango,
first, if it's Tango 1 involved in this,
okay, yeah.
and second,
I usually in
there's also COP1
I'm going to be able to answer one part of your question, and that's by not giving you a real answer. How do I do this? I usually say this, that it's somebody else's problem. You know, my job is in this life to be able to explain how collagens are exported from the ER. And this has been the problem of cisternal maturation, right? This is the whole business of, this is what rekindled the interest in cisternal maturation. Meaning, how do you export such big structures across the Golgi? So there are two ways of looking at this. When this structure fuses with the Golgi, the first cisterna just keeps moving forward, and what you're doing is you're moving things. Okay, so that's your cisternal maturation. Why? Because there are no tango 1-like molecules at the Golgi. So you don't need, once you've left the ER, you just keep moving forward. You don't need to resort these structures again. This is one way of saying it. Unless someone comes up with a tango 1-like molecule at the Golgi, which does the same thing again, I tend to think that it is highly unlikely. We haven't found anything. But there's always a surprise. Yes?
I presume that when you say that they try to see the vesicles containing collagen, is some sort of imaging that if this structure is the real one, they would have seen something similar to this.
Sure.
They wouldn't have seen the vesicles, but they would have seen this loop in the channel as you described.
Yeah, but you know, the thing is that these proteins take forever to leave the ER. And it is just not very, very easy. We thought we had GFP labeled collagen if we were making movies. It turns out GFP is cleaved from collagen because you can put it at the N terminus or at the C terminus, and both of those termini are processed. So we were watching GFP for a while. You know, so I think one has to be a little careful. Yeah, I'm really late. So can I move on to the next one? Yeah. Yes. Well, all I can tell you is that they are recruiting vesicles that contain urgic 53. So there are two ways of looking at this. One is vesicles coming back from the urgic. The alternative is, as you're forming COP2 vesicles here, they become uncoated. They contain urgic 53. You just rapidly recruit them. It's like before they get so far, you bring them back. We can't rule out that possibility at this stage. Yeah. But this is an important point. Okay. So anyway, so that was about the collagens. But you might ask, all right, so perhaps we know something more about the collagens now. What about chylomicrons and VLDL? So remember that our cells also secrete these huge lipoprotein particles. Okay, so what are these? These are basically lipid droplets. So remember, a lipid droplet will form in between the outer and the inner leaflet of the ER. And if they translocate to the cytoplasm, they become lipid droplet. But if they flow into the lumen of the ER, they are then coated by proteins called ApoB and many others, as shown here. And they become proteolipid or lipoprotein particles that are secreted. These lipoprotein particles are secreted. They go outside, and their job is to collect dietary lipids and deliver cholesterol and dietary lipids to various tissues. Okay. So you need these absolutely for maintaining lipid homeostasis, right? So there are many, many, many heart-related issues that come up because of defect in cholesterol levels outside and because of defects in export of these particular molecules. So how are these big molecules? They can be 200 nanometers in diameter. How are these molecules exported by the cells? So Antonio in my lab did something, you know, I don't like people sitting in front of the computer and just staring at the computer all the time. I like people pipetting. Maybe that's why I don't spend too much time in the lab. So you know so this is our tango gene, right? 1907 amino acid long protein that it produces. So Antonio found that there's another protein in the human genome which had been called MIA2 by some of the people in the literature. And what it is about 500 amino acids smaller than tango 1. It contains an SH3-like domain in the lumen. It doesn't have the coil-coil domain of tango. It has a transmembrane domain just like tango. It has two coil-coil domains like tango. And it has a proline-rich domain like tango. So it's smaller, but it has similar structure. Okay? So we reasoned that perhaps this molecule, alone or in combination with tango, might just be involved in export of chylomicrons and VLDLs. So is there any truth in that hypothesis, or is this hypothesis correct? So first, Antonio goes and looks at the distribution of these small. So tango is present in almost every cell type that we've looked at. Okay. Tali, which is tango-like, so that's the acronym for this new protein. Tali is expressed in liver, kidney, pancreas. Oh, I don't know what PR stands for. Small intestine is shown here. Okay. ApoB, which is absolutely essential for the export of these so-called apolipoproteins, right? If you don't have ApoB, you cannot leave the ER. Okay? This is expressed only in liver and in small intestine. It is, in our hands, also found in heart. I don't know the significance of this at this stage. Okay? So this told us that these proteins are present in the right cell type. So could these proteins be involved in the export of ApoB-containing lipid particles? So the experiments then are relatively easy. Okay, so what do? You, with CRISPR, knock down tango. When you do that, you don't affect the levels of Tali. Then you go with CRISPR and knock down Tali, and thankfully you don't affect the levels of tango. Now we have the parental cell line, tango depleted cell line, and Tali depleted cell line. And we can now ask, do they have a role in the export of these particles? So the approach is a bit tedious in the sense that it just takes forever to do these experiments. So you have to seed the cells and do various things to them for 21 days. And then after this period, you add oleic acid. This is to induce these so-called lipid particles. You wait 5 hours, you collect the medium, and then you process it. Now, a lazy person like me, I would just order an antibody to ApoB, collect the medium, and do a Western blot. But Antonio, hardworking man, decides that he was just going to collect the medium and sequence. He wants to do a secretome, basically. He runs a gel of the material collected from the supernatant of parental cells, tango-depleted cells, and Tali-depleted cells. And it just, I mean, the gel isn't the prettiest, but it makes a point. You see clearly there's one band that's present here that's highly depleted in these cells and virtually absent in these cells. Okay? The rest of the material that is secreted by the cells is more or less the same. So he cuts out this band and sequences it. And the sequence turns out to be ApoB. We got so many peptides. So I told you earlier that without ApoB, you cannot secrete these apolipoproteins. Okay? So it showed us that tango and Tali more than tango were required for the secretion of ApoB particles. Okay? Now, this can again be done by the lazy approach that I told you earlier, and that is to get an antibody and just do Western blots. So tango depletion affects secretion by about 50%, and Tali depletion affects secretion by about 80%. Okay. So this told us that these two proteins work together to help export these spherical structures. Remember, collagens are rod-like structures, whereas these are round structures. And of course, being a cell biologist, you want to confirm this by microscopy-based approaches as well. So here is looking at a cell, and this is, of course, done in many, many different cell types. In the nucleus, the green is the lipid part of this lipoprotein particle, and the red is the protein part. So what you can see is that the red and the green have assembled, but they're arrested in the ER, meaning that there is no defect in the assembly process per se. But once the structures have assembled, when you don't have tango and or Tali, you have a defect in their export. So this told us that tango and Tali both work together to export chylomicrons. I should make one point here. The Tali doesn't seem to have a role in collagen export. We've only tested collagen type 12. The problem is these proteins are expressed in different cell types, so you have to work with cells where the right collagen is expressed along with the molecules. So at least collagen type 12 does not require the function of Tali, and this is published. We published this last year. Okay. So I told you that tango assembles into rings in cells that secrete collagens. Does it assemble into ring-like structures in cells that express and export chylomicrons or VLDLs? And the answer is yes. So I'm showing you many different pictures here. Sometimes we can see a clear ring morphology. Sometimes it looks a little skewed. We don't understand that. And I'll try to get to this again in about 2 or 3 slides. So this tells us, this is again a very good confirmation that tango has the ability to assemble into rings, and then from the middle of these rings is the structure that's going to grow out into the direction of the Golgi, whether it's by vesicles or by this tunnel that I mentioned. We don't know at this stage. Okay. Now, soon after this story was presented, I've been presenting this for about good 4 or 5 months. It turns out that a paper appears from Yiheng Yu's lab at Tongji University in China that MIA6 is basically, for the sake of simplicity, is tali. Okay. The knockdown of tali in the mouse results in a defect in VLDL transport through COP2 assembly. So again, providing the confirmation to our work, which was done based on tissue culture-based analysis. Okay. So now, being a biochemist, or claiming to be a biochemist, I'm here amongst the giants in biochemistry, so I have to be a bit careful. But as Tom said earlier, as Roger said earlier, you're not that old yet, so I have time. Okay, so Anu in my lab spent good 2 years trying to purify tango. It's a hard protein to purify. Remember, it's huge, and it has this weird membrane topology that made it very difficult to purify. But ultimately, she was able to purify tango. This is the starting material. She was being able to purify this, and the idea is why we want to look at the structure, and we want to understand its ability to polymerize into rings that I showed you that are visible at the microscopy level, at least. So we've been able to purify it. And then we started a collaboration with a young investigator in London by the name of Giulia Zanetti. And this is just 2 days ago I got these pictures. So we are looking at the structure of Tango 1, purified Tango 1 in detergent. And I have a feeling that it assembles into a structure, into a small ring, but not perhaps a full-blown ring. So you can see ring-like morphologies. And you might say, well, no, you don't, Vivek. In some cases, it looks like parallel lines. And I'll tell you, this is very meaningful. So we see there's a structure. Okay? It's much smaller. There's a hole in the middle. But there seems to be some structure here. And we are now ready to do this, look at this by cryo-electron microscopy. So we'll have a very good image perhaps in about 2 to 3 weeks. Okay? This is just literally 2 days ago. So we see these structures, and so they are A, smaller in size, and B, sometimes they appear to be running in parallel lines. And this makes perfect sense to me, because A, they are devoid of COP2 coats. This is just purified Tango 1. So when we go and look at Tango's organization in whole cells, when its proline-rich domain is removed, so it cannot bind to COP2 coats, what we find is that, I know this is highly pixelated. Again, I got this picture from my postdoc Ishir, and I wanted to show it to you because of the microscopy data I showed you in the previous slide. You see it appears in the form of these long parallel lines. So our thinking is that Tango would like to run in a straight line, but when it binds to COP2 coat, it forms a ring like so. And this is the area then is marked for the growth of this structure. Okay. Anyway, this is perhaps a bit too preliminary for you, but I'm excited about it because I think it makes a lot of sense. I think this is the exciting part. Once it turns out to be the way we think, you say, okay, so we knew it, you know. So, yeah. So, you know, you can see that we can now do these kinds of experiments. We can remove this domain, we can remove this domain, we can express it in cells, and we ask what happens to the structure, and then we can purify those proteins and do the same experiments in vitro and look at the structure. Okay, so it helps us understand how this structure, how Tango assembles into these larger structures. Okay, so I think I've told you today that there is a Tango, there is a Tali, and it turns out that these proteins come in many, many spliced variants. So it used to be just CTH5. Kota identified that when he was in my lab, but it turns out now we are up to 16. So it's going to be a little bit more complicated, and I think the reason why we have so many is that when Tango binds to the COP2 coats, having one domain bind, one proline-rich domain bind to Sec23 might not be sufficient. This might answer your question. You need many, many, many of these proline-rich domains. It has to be a polyvalent interaction, and that polyvalency is provided by having many of these proteins which interact with Tango. Okay, so Tango not only forms a homodimer, it also oligomerizes with this. So how this affects the assembly of this reaction is something that we are also trying to address. Okay? So anyway, so these are the so-called proteins that assist. So certain cancers, by the way, certain cancer cells, so this is the work of Anja Bozarov in Germany. We haven't done this. So she finds that certain cancer cells export this part which is called MIA2, which is basically the SH3-like domain and part of the luminal domain. Okay? And the idea is that when these proteins are secreted, they will bind to collagens, and when they bind to collagens, you cannot assemble ECMs. When you cannot assemble ECMs, the cells have the capacity to to migrate. So this is the angle that Anya is trying to push. If this happens, then there should be a protease in cancer cells which can cleave here. And this is something that we are trying to find, whether there really is a protease that is not expressed ordinarily or not active ordinarily. And when I was visiting Harvard, Tom's colleague Bjorn Olson told me that tango, in fact, is phosphorylated at tyrosine 85, which is just above here. It could very well be that the phosphorylation of this tyrosine allows tango to interact with the cargos, or perhaps plays a role in this processing, and this is something that we are trying to address. Okay, so this is the lab, anyway. So this is the tango team. Ishir, these are the postdocs, Ishir Raute, Antonio Santos. Christina was a postdoc in my lab, and she's just moved to Oxford. Patrick Ullman was a postdoc, and he's running his own show at University of Innsbruck. Anu is a PhD student, my only one. Please, if anybody wants to come to my lab, to keep her company. Supported by two wonderful technicians, Marge and Maria. Fred Bard is at this position and in this place. Kota is in this capacity at Tokyo University. So I think this is the so-called tango part. And I would like to take another 10 minutes to tell you something even more exciting, if I might. So do you have any questions?
So your model assumes that tango is actually pushing collagen up, right?
Yeah.
So where does the
Energy.
force come
Oh, God. Everybody asks me the same question. So, no, it is very important, and I'm hoping that we will have an answer soon. So what we have done is, again, Anu has done this. So Anu has been able to put apex at different places along the molecule. Apex is a way to label the nearest neighbor, right? So she's been able to put apex here, all along the molecule, and we have identified lots of new proteins. And we are testing. So we've tested 7 of those 30 or 40 proteins that we identified in the lumen alone. And they all seem to have a role in export of collagen. We don't know why they are involved, but this is exactly the kind of question we would like to answer. One possibility is the assembly of collagen from being monomeric into trimeric might just be sufficient, but I don't know. I don't really have an answer. That is a very tough question to answer. Huh? No. In our hand, we looked at collagen type 7, but the other guys looked at many collagens here. Sure. Which is what? No. So there is this other idea that the Sec23 can be ubiquitinated by KLHL12, and this might have a role. In our hands, we have not seen any role at all of KLHL12, and what is problematic there is the site that is ubiquitinated in Sec23 is not known. So unless you know that site, it is just basically, if you knew, then you could just go and mutate it and just be able to address it. In our hands, knockdown of KLHL12 has no phenotype at all. And even in Randy's experiments, all KLHL12 does is when you overexpress it, it marginally stimulates the export from ER to the Golgi. It's not essential. So their thinking now is that under normal conditions, tango is the guy that is secreting collagens, but when you need to have a burst of collagen being secreted, for example, in chondrocytes during development, you might have built-in mechanism to speed up that process. But, so that's all I can tell you about somebody else's work.
Tango really including tango, is that possible that tango senses the size of the probe rather than
So this is hard to, because here, so there's another group who is now trying to, they are going to come out and say that enteroviruses that are huge particles, right? They are exported from the ER. They also seem to require tango. So the question is it the size that is being recognized, or is it something specific, or is this a certain set of molecules? HSP47 is not the only protein that binds. There are others that bind too. We can't really address this issue that easily. So we should try to make collagen smaller and smaller and smaller. Collagens are very hard to work with. You realize the problem is they need to be able to assemble into trimers. If they don't, they are rapidly taken out for export and degraded. So this is the reason why we haven't been able to do this simple experiment. Simple people, but it's technically very, very tough. Yes. 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 Yes, I mean, I think the idea is that you control the time at which you want to hydrolyze GTP on SAR1. Because if you hydrolyze it, then it's going to try to cut. But if you're still in the middle, and you haven't grown the structure, and you hydrolyze GTP on SAR1, you could try to pinch while collagen is still in the middle, when you don't have a big enough structure to carry it. So I think you need to time it. And that's why this becomes this part. This is very important. You need to have this structure large enough for Tango to dissociate so that Sec1331 can bind to then promote the cutting reaction. Now, we are assuming that this cutting reaction is the same as that used for cutting a small vesicle. But what if there is something else? There's another component involved in this process. So, once we have these molecules, we're just basically building on this thing, and hopefully we'll get to it. But for the time being, the function is of Tango with respect to the coats is to time the event that brings 1331 to 2324, so you can regulate the timing. That's the mechanism.
What is the additional contact that you're
Oh, so Jim and I were having a great discussion about this thing. It's a pity he's not here because he really loves this. He knows all about this thing. He sat me down for 3 hours and we discussed this thing. And he said, there's one thing missing in your talk. And that is, and I think he's right. So if you're pushing something from one side, if it doesn't have an attachment, it's just going to do this. Okay? But if you have it attached, then you can direct it. Okay? So could there be proteins that bind? So the simplest possibility is integrins. Integrins bind collagens. Okay? So it could be that a type of integrin is bound to collagen, and that provides the sort of, this is how you hold from both ends. Collagen is pushing and this structure is attached, and therefore you promote. And there is a sequence that is known, there is a sequence that is known to promote this interaction. And we are trying to now make integrins without this particular, it's only about 8 or 9 amino acids. So with Reinhard Fassler again, the idea is if we make such a mutation in integrins, do we affect the export of collagens indirectly through this particular mechanism? So this is in the works. Yeah. Okay, so just quick. Oh. Okay. So. Sure. I was going to tell you about this quickly, about a process called unconventional secretion, but I don't think I'm going to have the time. Sure. Okay. Sorry, I was going to tell you about unconventional secretion just briefly. This is a 2 minutes. Yeah. Oh, you 5. So I was going to tell you about two things. People talk about how membrane trafficking doesn't really seem to have any obvious clinical relevance. So I just told you about collagens. Then I was going to tell you about mucus homeostasis. So our understanding of how mucins are secreted is really very bad, much worse than even the collagens. So we did, again, a genome-wide screen, and we identified lots of new proteins. And we call them PIMS, for proteins involved in mucin secretion. And it turns out that we can test our proteins not only in tissue culture cells, but also in patients. We can take cells from people with cystic fibrosis and delete our respective genes. And so cystic fibrosis patients secrete lots of mucins, and we can reduce the level. So I think this might turn out to be very important. So we are and these are not snares, these are not COPs, these are not RABs. So these are proteins that we focused almost entirely on the mechanism by which you bring in certain amount of calcium to regulate this calcium-dependent process. So that's for mucins. And then the part 2 was this business of unconventional secretion. So, the conventional secretory cargoes are insulin, neurotransmitters, et cetera, et cetera, et cetera. Well, it turns out that there are also lots of proteins that are secreted without the proteins getting into the ER-Golgi pathway. So this was first done by DiNarlo in 1988 when he cloned IL-1 beta, and he found it doesn't have a signal sequence. So how could it be secreted, right? So we were trying to, I never wanted to work on this. We were working on how you generate a Golgi that looks like this. So this is the experimental data. This is imagination, right? Or this is what you eat. So the question is, how are Golgi membranes stacked? And this has been a question that people in our field have been trying to address since, the 70s. And we still don't have a very good understanding. So there was a protein identified based on in vitro analysis called GRASP. Well, it turns out that you can remove it from the cells and it doesn't affect the structure of the Golgi. So we reasoned that, come on, it must have a different function. So we deleted the gene from Dictyostelium discoideum, slime molds. There's only one gene for this particular protein. Well, it turns out when you start these cells, they go through the development process, they make spores and a stalk cell. And when you put these spores back in normal medium, they sporulate, right? But if you do the same thing with Dicty that have lost this gene, they fail to sporulate. Well, it turns out they fail to sporulate because they fail to secrete a protein called ACBA, which is basically diazepam binding inhibitor, which is the opposite of what Valium does, okay? It's a very important protein. It's highly conserved. So this particular protein of 9 kilodalton we found was not secreted by the cells. When we looked at the sequence of this protein, it did not have a signal sequence. So this made us think, could GRASP be involved in unconventional secretion? Because this protein cannot enter the ER. So the way we do these things, instead of working with Dictyostelium, which is a very difficult system to work with, we started working with Saccharomyces cerevisiae. We came up with an assay to measure ACB1 secretion, which only happens upon starvation of cells, not during growth. And you have to be careful that when you study this process, you don't want to lyse the cells, because if the cells lyse, these cytoplasmic proteins would be leaked and there'll be no signal. So Amy in my lab spent many years trying to get this assay to work. She has this. She can now measure biochemically ACB1 secretion only upon starvation. And this is GRASP dependent, as shown here. So it turns out GRASP is required for ACB1 secretion, interleukin-1 beta secretion, anything that is going out of the cells unconventionally. So this becomes the first protein in this particular pathway. But we also found that ACB1, before it's secreted, is always in a membrane-bounded compartment, meaning there is a structure, there is a membrane that hosts it. And the question has always been, what is that membrane? Well, we identified a structure, as shown here, by fluorescence microscopy in yeast. We can look at this structure by microscopy, and this is what it looks like. So it looks like it is composed of two kinds of membranes. A saccule, which contains lots of little vesicles. Okay? They contain the cargo. So ACB1 is in small dots, and GRASP is in these big dots. And what we have come up with is a scheme by which ACB1 is somehow collected into these structures that we call cups, compartment for unconventional protein secretion, which assembles from bits and pieces stolen from the ER, the Golgi, and the endosome. So the cells assemble this structure. They use it for secretion of this particular molecule. And then when secretion is no longer needed, the structure is dissolved, meaning it fuses with the ER and everything is returned to the compartments where the membranes were stolen. So the cells are really very smart. So it turns out that this is not just for ACB1. More recently, we've done a secretome in yeast to identify what other proteins are secreted. Turns out superoxide dismutase follows exactly the same pathway. And it turns out that many of the proteins that scavenge free radicals outside the cell, in yeast at least, they're all secreted by the common pathway. So this therefore becomes a bloody global Rothman-Sheckman Sudoff independent mechanism of protein secretion. And we are trying very hard. In yeast, the ease is that we can now do a screen, genome-wide screen, and we'll have all the components in a very short period. And we are trying to understand how this structure is formed, how cargoes get in, and how they're exported without this particular pathway. So that was going to be my bit on unconventional protein secretion, but I've been unconventional as always, and I've gone overboard. So I thank you all.